Biotech for Technical Education of Farming Society, Kota. Faculty of Veterinary Sciences and Animal Husbandry, Ranchi and Birsa Agriculture University. I cordially welcome you all to the second day of this 7 days training. This training is on a very innovative topic which everybody has appreciated. So the topic of this training is cutting edge innovation in veterinary science for a sustainable future. So before starting today's session, I would like to tell our participants a little bit about the organizers. So organizers are Vital Biotech. Vital Biotech is India's leading publication, research and training institute, whose goal is to tackle the world's most pressing problems which are related to food and agriculture. Whereas the Agriculture Forum for Technical Education of Farming Society, Kota, it serves as a spiral between the network of rural communities in India and other stakeholders such as government, development organization and corporate entities. Talking about Birsa Agriculture University, Ranchi. So this university was established on 26th of June 1981 and it was inaugurated by the late Prime Minister of India, Srimati Indira Gandhi ji. Primary objective of this university is to develop area-specific technology and manpower in the field of agriculture, animal husbandry and forestry for the development of Jharkhand district and the economic upliftment of the tribal and other backward classes of the region. The program and activities of teaching, research, and extensions are being carried out in this university among 7 faculties and 12 colleges. Besides this, university also runs a college of biotechnology, one center for agribusiness management, and 3 zonal research stations, farms, university farms, 16 KVKs, computer centers, central laboratory, etc. So the university is well equipped with all kind of infrastructure and required facilities. Talking about the science and animal husbandry, so the college was established on November 30, 1961. And initially it was a part of Ranchi University, but later on it became a constituent of Rajendra Agriculture University in 1971. And then it focused on the UG programs. Postgraduate courses were added in 1972 with further expansion by 1977. In 1981, it was incorporated into Birsa Agriculture University. So now it is offering UG, PG, PhD, diploma and certificate courses. So this college is also involved in research projects which are related to animal health, nutrition and conduct extension activities also. So now I would like to congratulate the entire organizing team and organizers for organizing this training program. And now coming forward and moving towards the today's session. So today we are highly privileged. We have such honorable dignitaries among us. So dear audience, it is a matter of great pride that today we have with us Dr. Pankaj Dhaka sir. Dr. Pankaj Dhaka sir is an assistant professor at the Center for One Health at Guru Angad Dev Veterinary and Animal Sciences University, Ludhiana. Sir has expertise in veterinary epidemiology, food safety, antimicrobial resistance, and farm biosecurity. Sir holds a PhD in veterinary public health from IVRI and MSc in veterinary epidemiology and public health from Royal Veterinary College, London. Sir has collaborated on several prestigious international and national projects, including serving as an elicitor for the WHO on foodborne disease source attribution. Sir is an expert consultant for WHO and he used to collaborate for tropical diseases. Sir has hold many international positions and Sir is a visiting professor at Ghent University, Belgium. Sir is editorial board member of multiple international journals and has received numerous national and international awards which recognize his contribution to One Health veterinary public health. Dr. Dhaka has published more than 58 peer-reviewed articles, books for textbooks, and has successfully led the multiple One Health focused research projects. And Sir coordinates for India's postgraduate diploma in One Health and also mentors students through International Student One Health Alliance mentorship program. So truly, sir, your achievements are the list is very long and it's not quite feasible to enlist all of them. So so with these words, I would like to request you to take over the podium and enlighten our participants with your insightful talk. So over to you, sir.
Yeah, first of all, thank you so much, Dr. Himani, for the nice introduction. And of course, to be honest, like I am also in learning phase like you all. So I'm sure we will have a good learning outcome. And again, I want to thank the organizer, Dr. Jitender, Dr. Dinesh, sir, and all of your team members for inviting me to this great platform. So today we will discuss one very important topic, that is One Health. So let me share my slides. So I will try to make you busy with some of the very important frameworks of One Health. Hmm. Okay. Okay, I hope slide is in this presentation mode.
Yes, sir. Slides are visible.
Okay, okay. Thank you so much. So first we will see in first half what is One Health, why it is important, and why we are discussing. And of course, you might have heard about this term long before, because of course the concept is not much new. But after COVID-19, this concept honestly is gaining much momentum, both in developing as well as developed world. And in second half of the lecture, I will share some of our Gadwasu leads in this area, so that you can get those who are new with term or concept, you can get some leads for your future research or like academic assignments. So let's start with very basic thing. What is One Health? Okay. So in very simple term, in layman term, uh, healthy animal, healthy human, and healthy environment can be only the sustainable way for our health. So it's a unified approach. So the approach is unified. It means there must be collaboration, and collaboration, of course, should be multi-sectorial and transdisciplinary. Uh, why we need to have the collaboration? Just to address local, regional, national, and global health issues. So remember, there can be different layer of issues, public health issues. For example, in Gadwasu, of course, we are more concerned about state level issues. Okay, then of course we used to think about national, then global. So it's depend of means at what level of collaboration do you need. Then the objective is to achieve optimal health outcome, recognizing the interconnection between human, animal, and environment. Environment means I mean like whole ecosystem. So why it is important? As I mentioned, if you want overall sustainability, you need to collaborate and you need to think about all these three components all together. Okay, for we will see. Okay, at least four very important domain where this One Health concept is really highly applicable. So what are the basic foundations? Here I mentioned three C's, but there is fourth C also. So we need to collaborate, we need to coordinate, and very important is to communicate what we are doing now. And the fourth C is generally capacity building approach. And now I will discuss with you four very important priority area of One Health where we are applying this concept very proactively. Of course, there can be many other areas also, but if we will put more focus on public health. There are four very emerging pressing issues. Uh, the first one is zoonosis, and I'm sure most of you might have heard the term zoonosis. So in very simple terms, these are the diseases which can be transmitted from animals to humans. So of course, most of us, not only most of us, like in India, if you will see 70% of our community is directly or indirectly in contact with animals, means farming community, we can say. You will be surprised to know like 75% of the emerging infectious diseases, which are either new or the diseases which are restricted in nature but now expanding its horizon, are from this subsection of infectious diseases, that is zoonosis. And third, 60%, sorry, 60% of all the infectious diseases 60% of them are zoonotic in nature. So you can appreciate how large the burden of these diseases are, and we must address them. If you will see, I told like zoonosis are the diseases from animal host. Okay, so there can be various species, as we know, like there can be wild birds, wild animal, stray animal even. You can see bats. I especially mentioned the bats because bats are very unique. They are mammals. They can harbor very high varieties of pathogens, and many of these pathogens can be very, very lethal to humans. And you will be surprised to know these pathogens don't cause any symptoms in the bats. Means the bats used to be asymptomatic, but under stress they can spill over, they can spread these pathogens. Then, of course, our farm animals with whom we are always means mostly in contact, either directly or through their food product. Then our pets also, pet animals also can cause many important zoonoses, including rabies, and then synanthropic, again very important, like rat, lizards, okay, especially rat, rodents. Rodents can harbor more than 53 pathogens. So I won't go in detail about individual, but we will now see why it is important. You can see this outline, timeline. You can appreciate after 1900s, okay, there are peaks in pandemics. Of course, one of the reason can be uh, honestly, like science developed much in much faster way after 1900. Okay, this discovery of virus, bacteria, and then theory of causation is this everything developed okay after 1900. But if you will see in after 2000 means in 21st century, I have highlighted downside. There are many pandemics means the organism or the disease is having worldwide distribution. It's not restricted to one or two country. So I have listed like SARS, swine flu, MERS coronavirus, Ebola virus, Zika, COVID-19, even monkeypox also we can add now. Okay, so in 21st century, even we are in starting phase of the 21st century and we have witnessed so many uh, pandemics. One of the reason you can cite like now we are having better diagnostic capacity. That's always good. We are having good surveillance. We can detect any like significant outbreaks very easily, like in case of COVID-19. So that can be one of the reason. But yeah, there are other reasons which I will enlist why there is so much surge in such type of outbreaks or pandemics. Again, you can see even unfortunately, okay, unfortunately, even on Times Magazine, time to time, these outbreaks make their space on the cover page. You can see SARS, bird flu, then avian flu again, then Zika, then swine flu, then COVID-19, our most notorious one, last one. If I will ask you, like, of course, the pathogens are always under transition, and we are always under biorisk. Means there are always is. But unfortunately, if you will see the list, there is much uh, the diseases which are emerging in nature. Of course, I have listed and I have already mentioned. So, okay, these are some of the diseases which we have witnessed in last like 20 to 30 years, okay, 2 or 3 decades. If you will see our efforts to control the infectious diseases, very limited diseases which we could have either only one we could have honestly eradicated, that is smallpox, and others, of course, we could have prevented to a substantial level. If you will ask me why we could have eradicated only smallpox, very simple answer because human is only the host. Even you will see others disease like many others, okay, like polio, measles, leprosy, tetanus. These are the diseases where humans is only the host, or maybe one or two hosts, okay, not many hosts. So always remember the diseases where only human is the host. It's very easy to target, but if any zoonosis is there with wider host range, it's very difficult to control. Okay, so always remember that's why it's really difficult to control the zoonosis. Even like Dr. Tedros, Director General WHO, time to time, okay, he used to warn there may be some disease X. Disease X means any scenario for which we are not ready. Okay, like COVID-19, in very simple example. So there is always danger. If you will see factors affecting emergence of infectious diseases, there can be many factors. Starting from these are some of the topmost factors which I highlighted. Started from deforestation, globalization, climate change, and loss of biodiversity. This is very, very important. So again, you can correlate climate change and biodiversity is directly associated with zoonosis emergence. Now come to the second important framework for One Health, that is food safety. As I told, like food safety, it means unseen threat on the plate. Why I wrote unseen threats because most of the foodborne diseases are not much noticed. Means first of all, we don't have proper surveillance systems in many of the countries to notice these outbreaks or incidences. This is one of the rough data by WHO. This is not based on actual surveillance, but yes, they hypothesized based on the available data or expert opinion. And what they found, around 4,22,000, that's especially children below 5 years of age, is associated with foodborne zoonosis. So this is really posing a huge concern. Again, the as I told, like the surveillance system is very, very poor to detect these changes. If you will see our farming, of course, the capita income is increasing in developing countries also, and there is huge demand for animal protein. And to meet this demand, we must have good output. Wood output means intensive farming. Of course, you might have noticed now that not now, even after 1950s, things are changing from extensive or semi-extensive to the intensive farming system. The main reason is economics. With less inputs, you are getting more outputs. Again, you will surprised to know this data, like per year, we are slaughtering around 70 billion of chicken. That is 10 times of the total human population. And this is 1 year data. So you can see how much burden is there on production side. And again, our global supply chain, you can see in this infographic, like if you are having salad in your burger or if you are visiting any good MNC restaurant, everything is coming from very, very different places. So a single hazard can contaminate the complete food chain. And yes, so we must understand, okay, in every context, the complexity of the food chain. Of course, most of the time, the food chain is not so simple what it looks. Okay, it's quite complex. And if you will see this graph, you will appreciate in 1900, in starting when our veterinary sector developed, we were more focused on individual animal. Then from 1950s, this herd immunity concept of herd immunity or herd health management came into picture. Sorry, it's after like 2000. Okay, now our focus is more on completely production chain, even starting from 1990s also. Okay. Uh, I hope you might have heard the term HACCP, that is Hazard Analysis and Critical Control Point. So that is like systematic approach which covers complete production chain, means starting from producer to the consumer. So that's very, very important. Now the third very important pillar or the framework for One Health to act is antimicrobial resistance, and this is again huge buzzword you might have heard everywhere. Okay, and the severity of problem is honestly huge. So I hope you know what is antimicrobial resistance means. In very simple terms, the pathogens are not sensitive for the available antibiotics. And of course, we are much more dependent, not only animals, in humans also, we are much more dependent on antibiotics, especially for infectious diseases. So why many? Okay, you might again you heard in news that this can be next possible pandemic. Yeah, now we are having data. This is data from one study from Murray et al. So in 2019, okay, to be exact, this antimicrobial resistance caused mortality of 1.3 million population. Okay, and as per the forecast by one British epidemiologist and economist, okay, John O'Neill, okay, he forecasted that by 2050 there can be possibility of 10 million deaths, and it will be equivalent to cancer deaths. Okay, so you can see if we won't put the checks and balances, this can be huge issue. Again, this infographic show like in 2020, if one this graphic of the human shows around 1 lakh death, in 2050 you can assume so much deaths. So in true sense, if we won't act properly, especially in developing countries, why developing countries? Because we are having limited options for treatment. If we won't act proactively, this is going to be a very big public health crisis. And what is the role of animal husbandry? Of course, we do have role. Again, this is the data from USA, United States of America. uh, Again, we don't have proper data, okay, because we don't have proper surveillance system to monitor antimicrobial usage in veterinary or animal health sector. But if you will see this data, you can appreciate like 80% of the antibiotic usage in animal. Okay, so this is again worrisome. And always remember, as I told, like pathogens can jump very easily. Okay, they won't respect any boundaries. Okay, so it is rightly said, resistance anywhere means resistance everywhere. And we must be very, very careful. Okay, we must be very proactive to first to develop to stop the development of this resistance gene, and if developed also, how to prevent or check them. Again, as I discussed, like our per capita GDP is continuously increasing, and because of this, yeah, the demand for animal protein is again increasing, and it's leading to intensive livestock production, and this in turn again uh, propel for more and more antimicrobial usage. And if you will see the projection also, there will be more demand, okay, and it has been like projected 50% more demand will be there for animal food by 2050. Okay, this is one of the forecast from again FAO. But the problem is we don't have proper surveillance mechanism. Okay, I'm talking about mostly developing countries. Okay, surveillance of antimicrobial usage. We don't have proper data how we are using, and where we can put the checks. So again, this is the area where we need to act. We will see why it is point of concern. Of course, we need antibiotics if the animal is diseased. Okay, for treatment point of view as well as for welfare point of view. But always remember, 75 to 90% of the antibiotics what you are administrating to your animal, it will enter into the environment in unmetabolized form. That means these antibiotic residues can put selection pressure on the environmental microbes, and they can acquire the resistance. Again, fisheries highly neglected sector. Okay, 70 to 80% of the antibiotics given to fisheries can excrete into the water. Again, agriculture practices like manures. If you are putting manure in your field, it can pose resistance. Okay, So this is complete chain. Always remember. Now the fourth component is environmental. Again, we have seen like zoonosis is affecting human, animal, then food safety, of course, to us. Then the third AMR, again, it can contaminate the environment. The fourth is very holistic, very broad component. Okay, environment. Whatever we are doing, it's directly or indirectly affecting our environment. So change in vector ecology, like you might have heard, many of the diseases, especially tick and mosquito-borne diseases, are affecting European regions. Because yeah, these previously these were the problem related with tropical countries, but now the vector activities has been noticed or increasing in temperate region. Okay, so again, this is point of concern, and this is directly related with climate change. Again, climate change is affecting our water supply and food supply, biodiversity loss, increased allergens, and of course, air pollution. Uh, ag- I want to correlate how environment and ecosystem can be related with emergence of zoonosis. So this is one concept, dilution effect. If you are having good biodiversity, it means you will have different host, animal host, and they will share various pathogens. If you will decrease your biodiversity, many of these pathogens can jump from their original or natural host to the intermediate host, or maybe in rare scenario directly to the human. So always remember, okay, biodiversity is very very important to maintain. So decreasing regulation services biodiversity will decrease the buffering. Buffering means it's acting as a buffer. Your biodiversity, your richness of the host species is acting as a buffer. If you will decrease it, that buffer action will went down, and of course there are high chances that the pathogen can spill over. Again, our bush meat and wet markets, okay, even in urban areas you will see wet markets. They can be very very big hot spot for spillover of the pathogens, because here we are mixing various species all together. And again, if you will destroy the ecosystem, there will be decreased availability of vital services also. It will, of course, if you will have lack of water and other things, it will compromise your hygiene. Now I will come to the domains where we are working. So just for your introduction, our Guru Angad Dev Veterinary and Animal Sciences University, it's a leading university, veterinary university of the country, and it's located in Ludhiana. And here, me and my team, we are belonging to Center for One Health. And previously it was department, then we upgraded as a school for public health and zoonosis. And in 2021, means very recently, we got upgraded as Center for One Health. So now I will show what we are doing. Of course, it will give you a idea. So first, I will share some of glimpses about the zoonosis, okay, where we are working. So in zoonosis, always remember what is the main issues. First of all, you need to know the prevalence, okay, whether the pathogen is there in your area or not. Second, if you know the pathogens, what are the important host or reservoir species of these pathogens. In this slide, again, you can see this I depict for Brucella. Brucella, I hope most of you know that it's highly endemic in the country, and the problem is there is wide host range, means the pathogen can be transferred from cattle, buffalo, goat, swine, even in you from your pet animals, dog, okay, mainly dog. Apart from that, you can see there can be many wild animal, even sea mammals also can be affected from this pathogen. So it's always important to know the prevalence and to have idea about the reservoir species. After knowing this, you must know what are the important transmission pathways, especially in your context. Always remember, for rural region there can be different transmission pathway, and for urban region there can be different. For example, in case of brucellosis, for rural region the main transmission pathway is direct contact with the infected animal. Okay, and but for the urban region, the transmission pathway is through contaminated foods. Okay, so always remember, okay, it is very, very important. If you know the transmission pathway, then only you can put intervention strategies. So this is our one recent study where we try to prioritize, okay, what are the important zoonotic diseases in Punjab. We followed scientific guidelines from CDC. We tried to develop our own ranking protocol. Then we carried out workshops with various stakeholders. Stakeholder, I mean veterinarians, academicians, and medicos. Okay, because of course we need to involve the human counterpart. Here you can see we in this side, like we tried to rank 15 important diseases. Okay, so brucella was number one. What was the criteria? You can see here. For more detail, you can go. This is published study. You can go to this publication. Okay, we broadly we try to target 7 very important parameters, and based on that, we found these are the 15 important diseases. Here you can see for like for fourth number, leptospirotoxo and HPI means they received equal weightage. Okay, so I won't go in detail. And again, I want to highlight one important pathogen, Coxiella burnetii. Okay, it's considered as neglected pathogen, but it is highly infectious, same as brucella. Here you can see one very catchy line: brucella and Coxiella. If you don't look, you don't find. It's true. And most of the time, we don't have good diagnostics also to detect this pathogen. But what is the specialty? Like brucella, it's having very wide host range. It can cause diseases in animal as well as in human. So this is one of our study where we try to find out risk factor among the animals, among the farm managements, and among the humans. Humans means farm workers. So again, I won't go in detail, but yeah, just for your knowledge, like in animals, we find large herd size. Why large herd size? For example, it's infectious disease. If the pathogen will enter, it can spread very easily. Okay, if you are not maintaining proper biosecurity, then exotic animal breeds we found like more sensitive. Old animal maybe because of less of immune function, then high reproductive disorder find to be directly associated. Farm management improper like disposal of placenta, poor biosecurity, environmental contamination, and occupational factors. Poor hygiene, of course. Okay, hygiene is always related with high risk of poor hygiene is always related with high risk of zoonosis, and raw milk consumption, same like brucella. So this is how it is very, very important. If you are going to detect any pathogen, you must have proper knowledge about the epidemiological risk factor, because it's if you know the risk factor, then only we can put the control strategies. Again, we were the part of one Brucella network program along with our 15 partners, and we developed some of the very important recommendations for brucellosis control. And we carried out some sort of, you can see here, cost benefit analysis also for vaccination policy. Like we found 8 to 11 times higher if we will invest in vaccination, because the problem with brucella in India is uh, because of religious concerns control animal. In countries, they used to follow the slaughter and test and slaughter policy. But in India, of course, we can't slaughter. So vaccination is one of the more important tool, okay, which we should focus. And luckily, now we are having at national level good brucella control uh, policy, mainly focused on vaccination. So these were our like some of the recommendations for the programs. Okay, like surveillance in animals, where we need veterinarians, then humans, then we need risk factor analysis, farmers awareness, development of the diagnostics. So that's very, very important. Luckily, for brucella, we are having some good tests, okay, which we can perform at field level like RBPT. Then yes, this is modeling, forecasting, then economic impact, then evidence-based prevention strategies and recommendation. Okay, so now come to the second component, food safety. Here also we are working, and our main target is to detect the sources of contamination, to carry it out risk assessment and intervention strategies. So here I highlighted some of the our work, recent work, of course, where we targeted foodborne, important foodborne pathogens. Here you can see like we targeting not only the pathogen prevalence, what are the virulent character of that pathogen and biofilm forming capacity. That's very, very important because in biofilm, what is biofilm? It's like community of the bacteria in very simple terms. They used to reside in groups, and of course it's beneficial for the microbes because they can be resistant to many like environmental stressors, but it is harmful for us because it's very difficult to remove the biofilms. And remember, if there is unhygiene in your food processing plant, there are high chances of biofilm formation if you are not carrying out proper cleaning and disinfection procedure. So here we tried to put some interventions by using lactic acid bacteria. We found very encouraging results. Like there was significant reduction of biofilms by using this bacteria. Even we tried to carry out some of the risk assessment, especially for aflatoxins. And aflatoxin is a very big problem. So being a tropical country like India, where we are having humid environment, and if the farmer is not storing the feed properly, there will be aflatoxin D1 in feed, and when the animal will consume the feed, it will convert to aflatoxin M1 very, very easily, and through milk it will excrete. And there, if you will go for proper surveillance study, I can assure you there will be really very significant amount of aflatoxins in our milk. So we carried out some of the studies based on prevalence as well as risk assessment, and even we tried to find out the association between aflatoxin M1 with human fermentation indicator. Here again we collaborated with our nutrition department, okay, to carry out this study. Again, now what in recent past we try to map out complete food chain of especially dairy sector. It is very, very important. If you will see, if you won't see the complete picture, of course there are high chances that you will get some of the biased result. So we try to map out the complete milk chain of like it's one of the milk producer of the country. The per capita consumption is number one, and farmers are very much aware. Again, we try to find out some of the important hotspot or critical control point in the food chain or milk production chain where we can target. And this is highly neglected. Very important. And regularly we used to carry out some sort of workshops. Okay, again it's a complete field. Okay, how to carry out the outbreak investigation in field. Okay, so again I won't go in detail. Now come to the third and very, very important, antimicrobial usage and resistance. And many, of course, many state veterinary universities are working on this. So what we tried to carry out this is one very exhaustive survey across the India with sample size more than 500 veterinarians. We carried out online survey and we found very interesting finding. Why there is misuse or overuse or underuse of antimicrobial antibiotics at field level. And again, downside you can see monitoring of antibiotic uses. This is the first study we carried out. Of course, this was like I can say convenient or targeted study. It doesn't reflect the true picture of the state. But yes, we targeted 30 animal farms and we carried out per month surveillance. Okay, means how much antimicrobial usage is there, which class of antimicrobials were there, and for which disease or cause they are using these antimicrobials. So again, there are very interesting finding. It's published study. You can go in detail. Okay, if you are interested in this area of antimicrobial resistance. And time to time, we used to carry out capacity building programs. Okay, we are very much involved uh, in capacity building programs to address the antimicrobial resistance. Again, we carried out one holistic review along with WHO, our partner, to see how in Southeast Asian countries we are using antibiotics and what is the status of resistance in food animal production system of this region. Now we started little bit work on farm biosecurity. That is very, very important. And this is a good scoping review. You can, if you want to see the association between farm biosecurity and antimicrobial usage here. Okay, this is just a brief overview about various factors, and remember these factors we highlighted from the evidences, okay, not from our side, okay, from the evidences generated from published literature, okay. These are the factors which are affecting both farm biosecurity as well as antimicrobial usage. Again, you can see gray, red, blue lines. Okay, they depict different things, and of course, we are having time limitation. Okay, so I won't go in detail, but you will appreciate. Okay, these factors. You can go through in detail. Okay, of this paper, and you will find how various factors can influence, okay, both of the practice, your farm biosecurity as well as antimicrobial usage practices, okay. And this shows these conditions are multifactorial, okay. We need different approach based on the evidences, okay. In simple words, like one size don't fit all to address this scenario. So farm biosecurity is completely related with one health, because if you are having healthy animals, then only you will get the healthy food. And of course, if your animal are ill, not only you, your environment also get affected. So always remember. So now even we try to carry out situation analysis, and we provided some of the recommendation for dairy farms of Punjab. And what we did, like, mm, if I will go to the farmer and if I will say that your farm is not good, means it doesn't make any sense. So we try to quantify, okay, their biosecurity level based on various parameters, okay. So if I am telling that you are scoring 80 score out of 100 in your farm, and your neighbor is scored like 95, then the farmer will be curious to know where he or she can put improvement, okay, at their farm. So always remember, like, quantification is always good, just like over marking schemes, okay. If you are getting good marks, it indicates that you are on right track, okay, more or less. So now we are doing a lot of work in this area, and this is really important, because if you are focusing on farm biosecurity, it will address your zoonosis part also, your antimicrobial resistance part also, and your food safety part also, and some component of environmental hygiene also. Apart from that, time to time, we used to map out various transmission ways. Very recently, we tried to detect like flies and role of flies and beetles, mediated transmission of the pathogens, then role of wildlife and pest in transmission of the pathogens animal in piggeries, and yeah, some of the recommendations regarding preparedness for public health threats, and how we can improve our surveillance system. This is our one of like review paper, and it's getting good attention in terms of citations and publicity. You can go through it. How we can like improve our surveillance mechanism to address zoonotic diseases. Okay, always remember, if you can't detect, you can't act. Okay, very, very simple. And fortunately, now we are having very good technological advancement, especially after COVID-19. Now it's completely era of artificial intelligence. So it's high time that we must revamp our surveillance system with all these technologies, so that we can detect and we can respond very, very efficiently in proactive manner. So strengthening of surveillance system must be the number one priority if you want to tackle the diseases. Again, just I want to highlight like we are the part of one very good project from DBT Pan India Consortium on One Health. We are carrying out such type of projects are very, very important because now we are working with multiple partners. There is good collaborations and exchange of data as well as knowledge. So you can see under this project till now we have collected around like Gadwasu Center I am telling okay 5,600 samples for various pathogens you can see for Brucella, tuberculosis, Cryptosporidium, Q fever, Strep typhus. So this really reflect like in collaborations we can do more and more work in more efficient way in more applied way. So that's one of the most important pillar of One Health, like to work in collaborations. Again, we are the part of one ICMR projects. Again, the beauty of this project is we are working with even with medical institutes and veterinary institutes and some of the agricultural institutes, and we are targeting slaughterhouses. Especially in India, you might be agree like most of the slaughter operations are carried out in unorganized way, not in under the organized way, and most of the time, okay, forget about the poultry, but if you will talk me about sheep and goat or piggeries, when the animal is having disease, they used to sell or refer it to slaughterhouses. So most of the time uh, slaughterhouses can be a very big hotspot for the zoonosis. Always remember. So we are targeting. You can see various list of bacteria, viruses, rickettsia, parasites, foodborne pathogens, and fungus among these slaughterhouse workers. And again, this is third very big project on which we are partner with many other institutes where we are carrying out training. Okay, so that's the part of communication. That's very, very important. You must communicate your finding. Okay, otherwise it will be restricted to the labs. Okay, so you must go to the field. You must talk to the stakeholders. You must tell them risk factors. You must tell them the interventions. So here we are continuously carrying out training programs among farmers and veterinarians about our findings on zoonosis, on antimicrobial uses, farm biosecurity, and animal welfare. Again, Government of India (DADS) is collaborated with FAO India, and program is ongoing for field veterinarians. Okay, and again, it's very very important about the field epidemiology. And this is, of course, at this stage, this is in initial phases, but I'm sure, like in coming time, it will give a very big boost to the field veterinarians how to deal with infectious diseases in more effective way. And these are some of our literature we developed, and I would be happy if you need. Okay, most of we are having in PDF format. You can access also. And yes, we are carrying out one postgraduate diploma program in One Health in distance learning mode. So if anyone among you are interested, distance learning mode means there are online classes. You can come in semester for 5 days and you need to give the exams. So this is 1 year program. If any one of you are interested, you are most welcome to join us. Generally, we used to call for this program in July, June or July. Okay. So time to time again, we are carrying out like training sessions for academicians as well as paravets, vets, and zoonosis keeper. So always remember, addressing zoonosis require collaborative approach. We must coordinate, communicate, and collaborate. And this is one of the very good resource. You can see right side of the screen. Previously, it was tripartite. Now it is quadripartite. Even United Nations Environment Program also joined this One Health approach. Okay, so this is one very good joint action plan guidelines. You can access this online if you want in more detail. Regarding India, now that as I told, now the things are gaining more and more pace, and we are having many multi-sectoral many projects, and I'm very hopeful like in coming time it will come more and more. This again, very recently, animal pandemic preparedness initiatives and animal health system support for One Health. These are in coming times, these are going to be a good surveillance platform. For like human side, now they develop integrated health information platform where they are regularly carrying out surveillance activities. Even our Nivedi also came up with very good program surveillance mechanisms by using the artificial intelligence. Recently, the government proposed for, I mean, not proposed, they have accepted, and the things are in has been very well channelized for National Institute of One Health. So I'm very hopeful, like in coming days in India, of course, it's in very good momentum. It will get more and more good momentum to address public health challenges. So with this, I am stopping my lecture and my deep acknowledgement for my faculty colleagues as well as our students who are working day and night for over many of our public health related, One Health related projects. So thank you so much. Thank you for your kind attention, and I would be happy if you are having any queries where I can put forward views. So thank you.
Thank you, sir. Thank you so much for this insightful lecture you have delivered, and our audience has indeed liked it a lot, and you have presented it very well. And I must say, the work you are doing and your team is doing under the One Health program and in your institute is very, very commendable. And you have shown all of your research and papers. It is very nicely presented by you. So thank you so much, sir. And indeed, we have a few questions. So the question is like, if first question is that what are the steps that are currently being taken by the field veterinarians to stop the outbreak of zoonosis and other diseases, and what are the preventions farmers or we need to keep at our level, like what we can do.
Okay, first of all, thank you so much, Dr. Imani, for your kind words. And yes, at current level, we need more and more communication, I will say, or awareness activities. Yes, at field level, veterinarians are doing wonderful. You might have, like, we witness LSD outbreak, lumpy skin disease outbreak. That time, we find that there is huge urgency of good surveillance and biosecurity protocols. Even Department of Animal Husbandry and Dairying, they are coming up, as I have mentioned, with FAO India good field epidemiology project, where they are aiming to train field veterinarians, as well as the most important tool we are having is farm biosecurity. Of course, it is gaining attention, but it's a complete, I can say, systematic science. Okay, we can put more and more focus to improve at field level. At farmer level, in Punjab, I know the farmers are having very good awareness, but again, in other states, I don't think the scenario is like this because we need more and more better awareness for that. Yes, universities must work with animal husbandry departments to carry out applied evidence-based research and to communicate them to the farming community.
Yes, sir. So another question is that, like you have told that the risk of aflatoxin uh, to the in the feed of animals, and then it contaminates the milk, and milk generally, and then human consume the milk. So how we can avoid it, and how we can just prevent this spread?
Oh, again, thanks, Dr. Imani, for pointing. This is huge problem. Okay, aflatoxin in milk is huge problem, and yes, everyone is trying to find the solution. The best solution is feed management, to be honest, because if you will control the point source, that is our feed, in which like generally aflatoxin B1 used to be there, which used to convert in M1. So in feed, first and foremost is proper harvesting and storage. And yeah, many of the farmers use now even using aflatoxin binders also. Okay, so this is one of means existing solution. But yeah, the real solution is feed management.
Okay, sir. One question which I would like to ask, like you have mentioned, like in India, we cannot slaughter the animals when we when we encounter any disease or any problem, and so we have to go for other measures like for vaccination and all. So how much effective these vaccination programs are, how much the vaccines are how much these vaccines are effective, and do they have any chronic effect on the health of the animals?
Hmm. Okay, again, good question. Okay, as I mentioned, we are not slaughtering the cattle. Okay, mainly cattle, and brucella is mostly prevalent in like dairy sector. Okay, and the problem is this: if we are not slaughtering most of the time. If farmer came to know that his or her animal is brucella positive, the first thing he will do to sell the animal. Okay, so the pathogen will spread very easily from one farm to another farm. First and foremost, yeah, better screening, testing is very, very important. And as you mentioned about the vaccination programs, so always vaccination is always important, but vaccination at large level to generate the herd immunity is very, very important. That is very, very important. For example, if you are vaccinating, if your farmer is not vaccinating, again, it's posing some sort of, if your neighbor, sorry, not farmer, if your neighboring farm is not vaccinating, it is always a hazard for your farm. Starting challenges, one of the very big challenges to motivate the farmer for booster dose. Okay. Because if you will put vaccine, of course, the immune system will generate in the animal, and because of that, there will be slight drop of production if animal is in milking stage. So again, the key is to communicate properly, to communicate the farmer about the science behind the vaccination. But yeah, after COVID-19, there is huge awareness, especially about vaccination, quarantine, isolation, and hygiene. So now the farmer understood some sort of biology of the pathogen. So that's good.
Yes, sir. Yes, sir. Now there is understanding among the farmer and among the common public. There is
Yeah.
understanding about the spread of diseases and these viruses, and the prevention which we should take care. And thank you, sir. It was indeed a very wonderful presentation, and we have got
Thank you. Thank you.
so many comments from the audience that the presentation is very good. So thank you for your
Thank
time.
you.
Thank you for presenting this topic. And I would like to tell the audience that audience can that people from audience, they can enroll in your One Health in your certificate courses which you have mentioned under your center and under your university. So I will recommend the audience to go and check out the website and details which sir has mentioned. So thank you, sir. Thank you so much for this interesting and fantastic presentation.
Yeah, thank you. Thank you, Dr. Imani,
Thank
Dr.
you, sir.
Jitendra, Dr. Dinesh.
Hello,
Yeah.
hello, hello.
Yeah.
I especially thank Dr. Pankaj. He accepted my invitation to present in the training program. It is a very excellent presentation and very nicely express their view and what he is doing presently. So I think it is very useful to field veterinarian also to how cope up with the genetic diseases in the field condition. Thank you, Dr. Pankaj, for accepting my invitation.
Thank you so much, Dr. Dinesh. And sir is always, sir remains source of inspiration for me. Okay, so thank you so much, sir, for inviting me and sharing this platform. Okay, so and thank you everyone. Okay, and feel
Thank
free to
you, sir.
interact me if you having any doubt anytime. Okay, not only on this platform.
Thank you, sir. Thanks a lot. So, dear
Okay,
participants,
thank
yeah. So, dear participants,
you.
now we'll move forward and we'll invite our next guest for his insightful talk. So, I would like to invite Dr. Mahesh. And before sir starts his talk, I will let our participants know a little bit about Dr. Mahesh. So, Dr. Mahesh is graduated from Veterinary College, Bangalore, and sir has done masters and PhD in animal nutrition from ICR NDRI Karnal. Presently, sir is working as assistant professor at Faculty of Veterinary and Animal Sciences, BHU, which is Banaras Hindu University, UP. Before joining academics, sir has 4.5 years of working experience in feed industries, including companies like Cargill and Camel, in functions like R&D, technical and marketing, focusing on ration formulation, technical services, feed additives, and pre-mix recommendations for designing nutrition-based profit strategy for compound feed manufacturer and dairy farmer across South Asia. Sir is a life member of Animal Nutrition Society of India, Animal Nutrition Association, and Indian Dairy Association, and sir has served as the editorial board member of Indian Journal of Animal Nutrition for 2 consecutive terms. Besides acting as a reviewer for a few reputed journals, sir has published over 30 research papers, edited a book whose title is Feed Additive and Supplements for Ruminants, and this book is published by Springer Nature. So in his present roles at BHU, sir used to teach UG and PG students on animal nutrition and guiding them for their studies and for their career. So at Livestock Farm Complex of this institute, sir look after small ruminant unit as section in charge. There is a lot to tell participants about the achievements of Dr. Mahesh, but still we don't have that much of the time. So without wasting much time, I would like to invite sir to deliver his talk. So over to you, sir.
Thank you, Dr. Himani, for nice introduction. So now I will be sharing my presentation. Kindly confirm if it is visible.
Yes, sir, it's visible.
And I hope my voice is also audible.
Yes, sir, you're perfectly audible and your presentation is visible.
Okay. So,
Yeah, thank you, sir. Over to you.
Yeah. So good evening, all the participants and organizers. So I welcome all of you for my presentation on the given topic that is related with distillers, dried grains with solubles as an emerging feed ingredient for livestock. So since yesterday, we have been seeing in this very important international training program. So some of the interesting presentations, 2 presentations we have seen. So unlike those 2 presentations, which were hardcore veterinary science presentations, so in this particular presentation, I will take you through some basic concepts of animal science with particular emphasis on animal nutrition. And specifically, we will be talking about DDGS. So that is distillers, dried grains and solubles as a prospective feed ingredient for ruminants. So the outline of presentation will be a brief introduction followed by dairy feed scenario in India and the novel feed ingredients available for ruminant feeding, including DDGS and its types and nutritional value, pros and cons of DDGS, so as reflected through literature insights, and some of the practical perspectives on the use of DDGS in ruminant feed formulations, followed by laboratory methods for quality control, and finally conclusion. So if you see the status of Indian dairying today, the statistics and status, as most of us are aware, that India is dominating in the world dairy production scenario since over 2 decades. We are the number 1 in global annual aggregate milk production, currently producing close to 240 million metric ton of milk, which represents 23 to 24% of the world total production. And so we have transformed ourselves from a milk deficit country to a milk surplus country to milk adequate country to currently it is a surplus. So we can see that the per capita availability of milk is currently 471 gram as against the recommendation of 280 grams. So that is a significant stride what India has achieved in dairy production. Whereas the milk trajectory by 2030, if you see, so that is expected to grow to 89 million metric ton, representing more than 25% of the world share. And by 2050, so there is an increase in milk exigency by over 50%. So whatever milk we are producing and consuming in the base year 2010 will be expected to increase by 50% by 2050. So what are the additional opportunities for Indian dairy? So one downside of Indian dairy production is that very meager or minimal export value. So the export of Indian dairy products are less than 1% of the total world exports. So that presents a huge opportunity for expansion of Indian dairying. So when every time we talk about Indian dairying or dairy production, processing, etc., feed and forages or feeding systems become central to overall dairy production, because close to 65 to 70% of the recurring expenses at farm level are related with animal nutrition. And in one of the industry surveys, it has been reported that animal nutrition solutions, here we can see, so this is the one solution which can have a significant impact on the overall sustainability of agri-food production. So that is the reason why we have selected specifically animal nutrition solution in this particular presentation to improve dairy sustainability. So how Indian dairying is transforming? So generally, if you see Indian dairy production, if you are discussing in global forums, so naturally people will say that Indian dairy production is number driven. So we have a huge number of ruminant livestock that produce a small quantity of milk per head. So aggregate volume will be more. So however, in the last 10 years, we have seen the significant transformation of productivity of animals. So here we could see first animal which produced 76 kg of milk in 24 hours. In February 2025, we have seen in Punjab, under Indian environmental condition, a Holstein Friesian cow has produced over 82 kg of milk in 24 hours. And in rightmost picture, we could see that Murrah buffalo in Haryana has produced over 32 kg of milk, setting world record. So these are not the outliers. So these are the examples that majority of the progressive dairy farmers are today having at least 10% of their improved stock, which are crossing 50, 60 or 70 liter of milk production. So that way we can say that Indian dairying is transforming into productivity driven dairying as against number driven. And maintaining these animals requires scientifically balanced rations for optimum production, reproduction and health. So this is one example to show how we are managing the nutritional requirements of elite modern dairy animals. So we could see that the number of rows here is probably more than 15 or 20. Today, the high yielding animals are managed with so many nutrients. So unlike earlier, we used to talk about only protein or energy or maybe crude protein, maybe TD and etc. But today, the high yielding animals, even if you talk about energy, that is fractionated into maybe minimum 3-4 elements, like maybe digestible starch, digestible NDF, non-fibrous component, non-structural components, carbohydrates, etc. So similarly, protein, which is expressed as a crude protein, to metabolizable protein. In between, we have rumen degradable protein, undegradable protein, digestible amino acids, etc. So this way, so scientifically balanced ration should comprise of so many elements in to manage and maintain high productivity of modern dairy animals. How the Indian dairy feeding systems and feed industry has evolved. So it would be interesting to know that how low productivity system to moderate to higher productivity. So here we can see that the feed production system and dairying has evolved from byproduct or home mix feeding system to today's scientific formulations, as we have seen in the previous picture. And improved physical farm or feed processing industry is coming up in a big way. So from simple home mixed mash feed to pellet feed formulation is becoming increasingly important, and which is common, at least in the dairy advanced states like Punjab and some other states. And improved feed segmentation like milking and non-milking and specialty feeds. So I would say that previously 10 years ago, so this concept was completely non-existing. So specialty feed for non-milking animals. So like for example, heifer feeds, calf starter and transition feeds, which at least even 20% of the progressive dairy farmers are aware of these concepts as on date. So greater consideration of KPI of cows, that is key performance indicators, production as well as health. So health, so earlier we used to focus primarily on milk. Today farmers, in addition to maximizing, they know the significance of optimizing the production. So once production is optimized,
I think there is a technical issue and sir has lost the connection.
okay,
Yeah. Yes. Yes.
aflatoxin controlled feed, therapeutic feed, including additives and supplements, etc. And major advancement in commercial forage processing. So like hay, silage and total mixed rations are not uncommon. And overall positive transition from average or imbalanced feed to more quality and premium feeds like balanced feeding, and sometimes also called as precision feeding, smart feeding systems, etc.
Dr. Mahesh, please share slide.
Sir, your slides are not visible now. Actually, in between you have lost the connection, and after that your slides are not visible.
Just a second. Give me 1 second. Oh, now it is visible.
Yeah, it's visible, sir.
Thank you. So we were discussing about how feeding systems have
Sir,
evolved.
slides are visible, but they are, yeah, now it's on slideshow mode.
Yeah.
Actually, earlier it was not on slideshow
Yeah,
mode.
it's okay.
Thank
So,
you, sir. Thank you.
yeah. So we can see that the summary is that the augmented milk productivity and feed efficiency. So feed efficiency means how much milk production is taking place per unit of input. So input is mainly feed by the cows with the latest technical know-how. So this way we can see that the feed industry as well as feed production as well as dairy productivity has improved in India over past few years. So whereas the ultimate reality is that the compound feed production or concentrate feed has not penetrated even up to 17%. So that in one way it is a low light, and in other way we can say that these represent a huge opportunity for quality feed manufacture in the coming days. So since the audience comprise of the mixed group including field veterinarians, students, veterinarians, animal science, etc. So this is one basic concepts. Everybody says that animals should be fed a balanced diet for producing optimum milk production. So it is very easy to say balanced feeding and balanced nutrition, etc. But practically it is not that easy. So what is the basic meaning of balanced feed? So here we can say that in a simplistic term, so animals require certain quantity of dietary nutrients. So those nutrients should be supplied in the diet which animal can consume in a day. So that is a simplistic meaning of balanced feeding. If you can fulfill all the nutrient requirements in the dry matter which a cow can consume in 24 hours, so that system can be called a balanced feeding system. So in nutritional terms, we say that macronutrients like carbohydrates, protein, and fats are macronutrients, and also micronutrients like minerals, vitamins, and feed additives. So combination of macro and micro will result into balanced feed, which is basically required for maintenance, production, and reproduction. And the basic components of balanced feed are both forages as well as concentrates. Forages include dry forage like wheat straw, paddy straw, etc., as well as stovers, and green fodders like leguminous green fodders like basim, alfalfa, lucerne, cowpea, as well as non-legumes like cereal fodders like maize, sorghum, bajra, and napier, etc. And the second component is concentrate, which is also known as compound feed. So what is the meaning of compound feed and how to prepare compound feed at farm level or at industrial scale? The thumb rule to prepare compound feed, so which is one of the major nutrient input for milk production, is that we need to include energy-rich ingredients, protein-rich ingredients, and energy protein intermediate, otherwise known as fillers. So energy-rich ingredients mainly include cereal grains like maize, broken rice, broken wheat, etc. And protein-rich ingredients include mustard cake, groundnut cake, soybean meal, sunflower cake, etc. So that means the byproducts of oil manufacturing. And in between, the fillers include wheat bran, de-oiled rice bran, chunni, etc. In addition, we also use additives and nutritional supplements like minerals, vitamins, etc. So this way we prepare compound feed, so which is basically one of the nutrient inputs to support milk production. And cost as well as nutrient values basically dictate the inclusion level of raw materials. So which could be either conventional or novel. So in this particular presentation, we are interested in some of the unconventional or novel ingredients like DDGS. How compound feed production is growing in India. So this latest survey shows that India is ranked fourth in a total compound feed production in the world, with a very impressive growth rate of CAGR of over 13%. 13% in India, representing both poultry as well as ruminant. Whereas paradoxically, we have that crop production and crop byproducts are available, growing at only 2%, whereas the feed is growing at more than 8%, and dairy industry put together is growing at 6% CAGR. So this is the one statistic or data that compels us to use unconventional ingredients or novel ingredients like DDGS, which we will be discussing in the coming slides. So what is the meaning of unconventional or novel feed ingredients for dairy production? So here we could see that the feedstuffs that are not traditionally used in diet formulation can be considered as unconventional or novel ingredients. So some of the examples include DDGS. So its nutritional value is variable depending on the grain source of used. So generally it is rich in crude protein as well as fiber, and it is obtained from ethanol or distillery industry. Second is maize de-oiled cake, so which is popularly known as maize DOC, which has become increasingly available in last 2 to 3 years in India, having protein value of up to 28%, which is again a distillery byproduct. And rice gluten meal, we have similar to DDGS. Gluten meals are the byproducts which are protein concentrates obtained after starch wet milling of the wet milling industry. And some of the other ingredients we have is Phalaris minor seeds, so which is a weed found in wheat feeds that can also be used as animal feed if available, with the impressive starch content of up to 50%, comparable with barley grain. And other any ingredients which are used by feed industry include bakery meal, sugarcane press mud, fenugreek seed meal, cashew nut meal, coffee husk, palm kernel cake, daincha meal, etc. So if you are talking about DDGS, so the major source for DDGS in the recent days is maize. So if you see the maize or corn production and utilization landscape in India, over 60% of the total maize produced in India goes for animal feed. So which includes cattle feed, poultry feed, aquacultural feed, as well as pig feed. So how this corn is converted into DDGS in the process of ethanol manufacture. So this is the schematic view that shows how we process corn grain to get alcohol or ethanol as a main product, leaving behind DDGS. In a simplest term, we can say that corn is rich in starch to the extent of 60 or 65%. So when corn is fermented, this starch is getting converted into alcohol or ethanol through enzymatic process. And in this process, protein is getting concentrated. So along with these enzymes and yeast metabolites, etc. So this DDGS will be used in animal feed. So we can say that if we are using maize as a raw material, so we will get maize DDGS in proportion of 5:1. So that means for every 5 kg processing of maize grains, we will get 1 kg of DDGS. And if we are using rice for the production of ethanol, so the ratio will be 3:1. For every 3 kg of rice, we will be getting 1 kg of rice DDGS. So this is one example. So how we are seeing increased diversion of corn that is produced in India for fuel ethanol. So government of India has set an ambitious target recently that includes blending of ethanol in the petrol to the extent of 10 and 20% by in the near future. So that has resulted in we can see that in 2022-23, 2 years ago, the total maize which was diverted to ethanol production was 1.7%, which has increased to more than 20% in 2023 and 24 last year, and this year it is expected to touch up to 28%, and by 2030 it is reaching to 32 to 33%. That means approximately 1/3 of total maize production in India will be diverted and utilized for fuel ethanol production. So this will result into the tight demand and supply scenario, so which was -2% in 2022, and which is expected to be at -23% by 2030. So while this represent one opportunity that increased maize ethanol production will result in increased DDGS, simultaneously availability of maize or energy for feed industry will be becoming reduced. So that results in increased energy price compared to protein price. So this is the latest data that shows that how much is the availability and penetration of DDGS in animal feeds. So with the increased availability of DDGS over last 3-4 years, so we have 4 to 5% penetration of DDGS in the total compound feed production in India, which was almost absent approximately 5 years ago. Although we say we discuss much about DDGS in recent years, so DDGS is not something which is extremely novel or new ingredient. So if you see the global scenario of DDGS utilization in production, so it was used since 70s or 80s in countries like US and in European countries. Whereas because of the impetus for fuel ethanol production through corn in the recent days, so DDGS has become increasingly available in India. And to dispose of this DDGS from the distilleries, so obviously it is diverted to livestock industry, and that is the reason why we are more and more discussing about DDGS in recent days. So improved availability of DDGS year on year, which represents a futuristic novel feed resource for feeding ruminants. And so as we have discussed previously, that DDGS is easing the protein prices while proportionally rising energy cost, because DDGS is produced from the energy-rich ingredient like corn. So that results in shortage of corn for feed manufacturing, resulting in its higher cost. Whereas protein price is becoming comfortable or low with DDGS. So earlier we used to say protein is expensive, but today and in near future we will be saying that energy is going to be the primarily expensive nutrient in ruminant feed formulation rather than protein. So what is the nutrient composition of DDGS? So if you see the original corn grain and corn DDGS, so protein percentage in corn is 8.5% approximately, and the maize DDGS protein will be 27 to 30%, representing almost 3 to 3.5 times increase in the protein value. So obviously starch content will be less because most of the starch will be converted or consumed during the process of fermentation and ethanol synthesis. And relatively, NDF is also getting concentrated in the maize DDGS to the extent of 40%. And similarly, ADF also. And ash percentage is also. So ash represents mineral component. So DDGS is a rich source of minerals, especially phosphorus, sulfur, etc., compared to original grain. And importantly, fat. So energy component of DDGS rather than starch in the initial grain is mainly attributed to fat, so which will be about 9 to 10%. So similarly, we have rice DDGS also. Compared to maize DDGS, rice DDGS is very rich in protein, approximately 45%, with similarly reduced starch content. So relatively, compared to maize DDGS, rice is having relatively less NDF value, and it is also and it is also having relatively less fat content compared to maize DDGS. So in a nutshell, what we can say that grain to DDGS conversion ratio is 3 to 3.5 times. While this is in for nutrient values, this is a benefit. So this is also having a demerit in the sense that if the maize is having some toxin like aflatoxin, which we have discussed in the previous speaker also highlighted, that if the maize grain is having aflatoxin B1, for suppose it is having a 20 ppb, so in maize DDGS we expect 3 times higher, so it will be 60 ppb. So that is one of the negative sides of using DDGS, because all factors will be concentrated to the extent of 3 times. So this is again the same thing that corn is a rich source of energy to starch, whereas corn byproduct, that is DDGS, the energy is primarily because of digestible NDF as well as fat. So as a ruminant feed formulator or dairy producer, we always compare the nutritional value of available ingredients, conventional as well as unconventional ingredients, which is based on nutrient value as well as price. So if you compare maize DDGS with all other conventionally available ingredients, like for example, mustard cake, mustard oil cake, soybean meal, cottonseed cake, cotton DOC, etc. So we can see that the price per kg protein has been significantly low in both DDGS, the maize DDGS and rice DDGS compared to even mustard cake. So mustard cake and mustard DOC, for example, are considered cheapest source of protein in India, while which is true for mustard DOC. But mustard cake, which is having about 6 to 10% of residual oil, whereas maize DDGS is also having residual oil. So maize DDGS is economical compared to mustard cake, and price per kg fat also. So we can say that it is economical in terms of providing both economical protein as well as fat for animal feed manufacturing. What makes DDGs attractive in feed formulation for ruminants? So economic delivery of protein and fats, as we have seen in the previous slide. relatively greater RUP percentage, that is rumen undegradable protein or bypass protein fraction found in the protein of DDGS compared to other conventional ingredients. Improved palatability, especially of maize DDGS with no anti-nutritional factors, because most of the unconventional feeds, if you take one or the other anti-nutritional factor, will be present in most of the unconventional ingredients, whereas with DDGS, a good quality DDGS is known to have no anti-nutritional factor. Rich source of essential minerals, especially phosphorus and sulfur, so particularly useful to reduce metabolic disorders with higher sulfur content. Source of non-forage fiber, useful for high-yielding formulations, because as we target improved digestible NDF, which enhances rumen fermentation and avoids any possible lowering of ruminal pH, especially during early lactation. Coarse powdery texture of DDGS mixes well with the other ingredients. So preparation of on-farm TMR becomes easy when we incorporate DDGS at farm level. And naturally, the increased human edible feed conversion efficiency can be obtained by cows when we are using DDGS, because DDGS is being extremely poor to no human edible value. So these are some of the factors which makes DDGS attractive for consideration in ruminant feed formulation. So with brief introduction about DDGS, now let us take up some research trials which examined the practical value of DDGS in dairy feeding. So this is one study from NDRI that considered use of rice derived DDGS at 50 to 75% replacement of oil cakes. Oil cakes combining groundnut cake and mustard cake were replaced by 50% in T1 and 75% on the isonitrogenous basis in T2 group. So this basically we can see that resulted in improved dry matter intake at higher inclusion, that is 75% replacement, which translated into improved milk yield. So we can see that in control, the milk production was 8.8 kg. So which was almost, we can say that close to 1 liter increase in milk production was noted with 75% replacement of this conventional oil cakes by rice DDGS. So which can be attributed to improved dry matter intake. So why DDGS or such fermentation byproducts improves dry matter intake? The reason behind this is that this DDGS also contains some of the yeast metabolites or residual yeast and other bioactive protein and components, so which are thought to be increasing voluntary dry matter intake in animals. So that resulted in improved milk production. And beneficially, there was no difference in any milk composition. So this way we can say that farmer, if he considers to replace groundnut cake or mustard cake or any conventional cake with DDGS, he is certain that it is certain that he will be getting improved milk realization at farm gate level. So similar to DDGS is one more byproduct of wet milling manufacture is rice gluten meal, replacing oil cakes. So similar to the previous, here also we replaced 50 and 75% of groundnut cake, 50% replacement of groundnut cake with rice gluten meal in T1 and maize gluten meal in T2. So which resulted in a non-significant difference in milk production between control and rice gluten meal, whereas maize gluten meal resulted in significantly improved milk production compared to control. So similarly, there was no effect on any milk composition. So this way we can say that DDGS as well as gluten meals could improve farm economics. So because with the same or with the similar input cost, if you are improving the production, so dairy efficiency as well as economic efficiency at farm level can be augmented. So this proves that DDGS and gluten meals could be economical and positive value proposition for dairy feed producers. So we have considered individual studies. So this slide shows the effect of inclusion of DDGs in ruminant feed in a meta-analysis. So meta-analysis means most of you may be aware of meta-analysis. That so each meta-analysis considers at least 50 to 100 research papers with more than 100 data treatments. So the conclusion from the meta-analysis can be said that it is almost universal, or we can say that it is concrete conclusion. So in first experiment, we can see that Malik and co-workers recently reported with dairy as well as feedlot cattle, inclusion of DDGS resulted in no effect on dry matter intake as well as methanogenesis. Whereas in case of small ruminants like growing sheep, meat sheep, 20% inclusion of DDGS resulted in increased digestibility, increase in carcass weight, and beneficial meat flavor. So, which is again, which proves that DDGS inclusion is useful for fattening sheep. In case of growing and feedlot cattle, so this Torres and co-workers in Mexico recently reported that high-level inclusion to the extent of 50 to 60% inclusion of DDGS resulted in favorable response on carcass parameters, including meat fatty acids like beneficial health-promoting fatty acids like conjugated linoleic acid was increased along with linolenic acid as well as total polyunsaturated fatty acids with a reduction in saturated fatty acids like myristic acid. So this shows that the functional value of meat can also be improved in inclusion of with the inclusion of DDGS in meat animals. While in growing cattle and heifers, it was shown that body weight gain, that is average daily gain, and final body weight were increased with the inclusion of DDGS. And interestingly, it was also found that DDGS could replace some forage component in the diet because in fattening ration, typically the fattening diet comprises of over 70 to 80% or maybe sometimes 90% of the concentrate in relation to forage. So DDGS usage may offer an opportunity to improve the rumen fermentation and avoiding possibility of acidosis with the help of digestible NDF contained in it. So how can we leverage the most of DDGS in practical dairy diets? So if you see that in a typical dairy farm, we have calf, heifer, dry pregnant far-off, close-up transition, fresh lactation and other feeds. So the suitability of DDGS is very negligible, or we can say it is rather unsuitable for calf formulations because amino acid profile of DDGS is low in lysine, and lysine deficiency may result into stunted growth in case of calves, poor protein utilization, and NDF or fiber rich, and probably the contamination of DDGS with aflatoxin may result in the untoward effect in case of ruminant calves. Whereas in case of heifer as well as the dry pregnant far-off transition cows, so we can say that DDGS is more suitable because heifer and dry pregnant stage animals generally requires less energy dense formula and relatively greater NDF is required, sometimes 40% or more than 40% also. NDF is suggested for this heifer as well as dry animals. So in this particular scenario, DDGS suits well in the formulation. So more than this close-up ration, we can say that its inclusion is more beneficial. So in addition to other factors like protein and digestible NDF, sulfur content is particularly helpful for close-up formulation because in close-up for transition, minus 21 days of parturition, we need negative decadration. So dietary cation anion difference should be negative. So that is about minus 100 to minus 150 milliequivalent per kg of dietary dry matter. So since sulfur content is higher, so which results in negative decad. So value will be negative, which otherwise we call it as anionic diet, which is useful to decrease the incidence of milk fever in transition cows. And similar is the case with the fresh lactation. Digestible NDF is useful to decrease subacute ruminal acidosis, easy to blend with other components in the TMR, and alteration in the physicochemical properties of milk. So some of the research also showed that increased inclusion of DDGS may result in some physicochemical variation in the properties of milk. So that could be one line of caution we have to take, especially when we are using DDGS in the fresh lactation. And the last case is particularly applicable to Indian condition. Any least cost or budgeted feed, like for example, BAS type 2 formulation, so which is very much suitable. We can say that economic delivery of both protein and fat results in the more suitability of DDGS in budgeted feed formulation, which is true for both rice DDGS as well as maize DDGS. So DDGS offers opportunity for feed cost savings. So whatever we discuss in animal nutrition, unless that results in improved performance at low cost, so we cannot say that the technology is useful. So this is one simplest example I have taken. How can we replace soybean meal, which is conventionally used protein meal in high yielding formulation, by at least 3%. So 8% is the soybean meal inclusion in first formulation, and 5% is the inclusion of DDGS in the second formulation. That means about 5% replacement of conventional meal. So soybean meal resulted in the difference of about 310 rupees of feed cost savings per ton of compound feed. So when we formulate BAS type 1 compound feed for ruminants, that is one of the reasons why, although we don't know much about DDGS, so just like a dark horse, so ultimately it is winning. So that way, some people describe DDGS as a dark horse in feed industry in current context, because with the improved nutritional value, which is also providing opportunity for feed manufacturers as well as dairy producers and veterinarians to optimize feed formulation for dairy animals. So these are the highlights or positive sides of DDGS. Now let us take up some of the limitations or demerits of using DDGS in formulation. One potential limitation is high moisture leading to high aflatoxin contamination, because most of the distilleries are primarily interested in enhancing ethanol yield. So to get more and more ethanol from the grain, so they may be processing whatever grains that are available in the market. So that means they may use best quality grain. They may also use some of the fungal contaminated grains. So in that situation, aflatoxin will be getting concentrated in the resulting DDGS, as well as the DDGS being a wet product. So if the drying is inadequate at distilleries, resulting in more than 16 or 17% moisture at the time of receiving at a feed mill or farm level, that could again predispose the DDGS for secondary mold development, leading to further aggravation of aflatoxin level. So put together, this results in health risks for both livestock as well as humans. So aflatoxin B1 will be converted into aflatoxin M1 to the extent of 3 to 6%, and excreted in the milk, which is known milk carcinogen. So it is always necessary to screen for aflatoxin when we are using DDGS. So even when it is unavoidable situation to use high aflatoxin DDGS at farm conditions, so what we can do is we can dilute this high aflatoxin DDGS with the fresh DDGS having low aflatoxin. So that is a practical strategy what we can do. And second major limitation is Maillard reaction or Maillard protein, which is getting concentrated in the DDGS because, as the name itself indicates, D refers to dried. So distilleries have to dry the DDGS, the byproduct, in order to make it suitable for transportation and further utilization by feed industry as well as dairy farmers. In this process, if the drying is inadequate or if the drying is too rapid and artificial drying resulting in Maillard protein generation, that results in dark coloration of the DDGS. Although color chart is not very much reflective of the quality of DDGS, but it can be considered that the more towards lighter color or yellowish or honey golden colored DDGS is having a better protein value as compared to darker or coffee colored DDGS. So this way, there is a more collaborative work is warranted among key stakeholders like industry, academia, collaboration, including feed industry as well as distillery industry, so that we can able to develop or we can able to streamline the quality consistently for usage by livestock farmers. So what are some of the methods to assess the protein quality of novel ingredients, including DDGs for ruminants? So generally in feed industry or today we can say that most of the protein is assessed as crude protein, which is nothing but nitrogen multiplied by 6.25, estimated by either Geldal method or NIR, etc. Whereas those ingredients which are having artificial, which have undergone artificial drying, so like DDGs, in addition to crude protein, we should also estimate, for example, available nitrogen, that is total crude protein minus ADICP, acid detergent insoluble crude protein. If the acid detergent insoluble protein is higher, that means available protein will be less. So that way, in addition to crude protein, we can also go for acid detergent insoluble nitrogen estimation. And also, if somebody is interested, this CNCP nitrogen fractionation is one of the latest methodology which researchers across the globe as well as some of the feed industry in India are also started analyzing feed protein for CNCP fractions, where fraction C would represent unavailable nitrogen, so which is also known as Maillard nitrogen or acid detergent insoluble crude protein, which should be as low as possible. So this way, in addition to CP, simply we can consider ADAN to know the practical value, or which is an indirect measure of availability or protein value of DDGs for ruminant feeding. So what are the factors to be considered or the summary of DDGs utilization? So aflatoxin, yes, because it is a artificially dried product as well as, and it is a fermented product with which is undergoing high moisture treatment. Aflatoxin issue is one of the potential threats associated with DDGs. Nutrient variation from grain to grain as well as distillery to distillery. That is one of the variations we have in today in DDGs, which is available in India. High moisture and mold contamination and mold growth is one of the negatives of DDGs. Lysine deficiency, especially for monogastric formulation as well as pre-ruminant formulation, and also high yielding formulations, especially in early lactation. And also when maize products are used at increasing level, for example, corn silage, corn grain, and corn DDGs. So if the corn products constitutes more than 50 or 60% of total TMR, total diet, so that could exacerbate lysine deficiency. So formulators should be cautious about balancing metabolizable lysine when maize products increase in the formulation to the extent of 50 or 60 or more than that. Poor protein utilization, especially in roasted or in maillard product contained in the DDGS. And from compound feed manufacturing point of view, there are some reports saying that because of the increased NDF content in the DDGS, proportionally, the pellet durability index of compound feed is compromised because PDA is inversely proportional to the total fiber content in the ration. So this could also affect PDA. Although nutritionally it may not affect much for a dairy cow, but for a commercial feed manufacturer, so this may be an important factor to consider. So with this, I conclude the presentation as DDGS, which offers some breathing space for dairy farmers, especially when there is a price volatility in the market and for dairy farmers and feed manufacturers when supply demand situation of raw materials are tight. And this way we can say that DDGS can be a prospective feedstock for Indian feed industry. And its usage is suggested when merits outweigh the inclusion cost. And any nutritional interventions at farm level cannot be considered as a substitute for good feed bunk management. So feed bunk management and feeding management, good management is always primary to any nutritional strategy. So with this, I thank all of you for your patient hearing. And I take the opportunity to thank the chief patron of this international training program, Vice Chancellor Sir of Birsa Agriculture University, and Dean Faculty of Veterinary and Animal Sciences, Ranchi Veterinary College, as well as Organizing Secretary, mainly Dr. Dinesh and Dr. Kullu, as well as the associates, that is Vital Biotech and AFTEF Society, for providing me this opportunity to share some of my views with this August gathering. So thank you once again, all of you. And if you have any specific queries, or if you want to share something, or if you want to add some input, so now probably we can discuss.
Thank you, sir. Thank you so much for your insightful talk. And yes, we have some questions from the participants, as we can see in the chat box. So participants have found your presentation very good, very interesting, and they were indulged in the presentation during the entire talk, and it is quite visible from the questions which we have got in the chat box. So we have a question like I'm merging 2 to 3 questions which are interrelated. So we have question like DDGS, it can be incorporated in poultry feed or not, or do it have any impact on the reproduction as we use it as a single protein source, and does it have any harmful effect because it has a high sulfur content? We get high sulfur content from DDGS, and how this sulfur can affect the metabolism of other minerals.
Correct. So first of all, thank you, Dr. Himani, for your compliments. And as I could see the questions also, first question is DDGs can be incorporated in poultry. Yes, in poultry, we have two challenges. One is quality of DDGs in terms of aflatoxin, that is primary. And the second one is protein quality in terms of protein digestibility. So the put together, considering both the factors, so as I'm not directly involved in poultry formulation, but as I have heard from the poultry experts that they are using this rice and maize DDGS to the extent of 3% in broiler starter and pre-starter formulation, and to the extent of maybe 5% or more in other formulations like broiler finisher. And in layers, as I have heard from the experts as well as literature, so there should be no problem with higher inclusion of DDGS in layers. So that is one thing. But in either of the case, so aflatoxin is a major issue. So we have to curtail, we have to stringent our procurement protocol to streamline and stringently check for aflatoxin, especially when DDGS is meant for poultry formulation. So second one is what is the impact on reproduction when using DDGS as a single protein source? So concern with reproduction primarily, in my opinion, stems from probably high mycotoxin contamination. So rather than any other component in DDGS. So if we can have a check on aflatoxin levels, so that should solve reproduction. And one more challenge with DDGS in reproduction is wet DDGS. If a farmer is using wet DDGS, that has to be disposed of within maximum a duration of 48 hours. So, which is a practical challenge. Especially, I have seen in southern states like Karnataka, Andhra Pradesh, etc., and in peri-urban dairying in, for example, in Kolkata, buffalo farmers are using DDGS and spent brewer's grain. So they report some of the practical issues with the reproduction. In my opinion, that is primarily related with poor storage, resulting in secondary mold growth and aflatoxin contamination. And third one is, yes, sulfur is harmful or beneficial. It depends on, as of now, we don't have a crystal clear database of sulfur profile of Indian DDGS samples. That too, from grain to grain, it varies from rice versus maize. So definitely high sulfur is not beneficial because sulfur level is restricted to 0.2% of the ration, and it cannot cross 0.5% even in the close-up transition ration. So this the answer is both yes as well as no, depending on the source and particular feed in which we are using. So similarly, high sulfur can affect the other minerals. Yes, that is true. And is there any adverse effect of DDGS on the digestibility of milk fat? Yes. Digestibility, no report has been found, and most of the report, even international literature, supports positive aspect of DDGS on digestibility, especially overall organic matter digestibility as well as NDF. And with the improved NDF digestibility, obviously milk fat should be improved. So that way, so there should be no effect on milk fat also. But because of the higher fat content, that too polyunsaturated fatty acids, some people opine that it may adversely affect milk fat. But literature supports that even 40 to 50% inclusion of DDGS did not affect milk fat, especially those reports emanating from United States. So next one is it easily available to Indian farmers? Yes, it is becoming increasingly the quantity is becoming increasingly available, and farmers are receptive and open for its utilization. We can see most of the peri-urban dairying, and especially in northern belts like Rajasthan, Punjab, Haryana, and including almost all states, including we can see southern states like even Karnataka, Kerala, Andhra Pradesh, Maharashtra, Gujarat, etc. So I have started using, at least in the feed industry, started using. And in northern states like Punjab, I have seen that farmers are also using DDGS, corn gluten feed, as well as rice gluten meal, and even corn bran and corn fiber, etc. All these distillery products have been included in the ruminant formulations. I think yeah, there is one more question. What is the shelf life of DDGS? So shelf life of dried DDGS, there is no worry as long as the moisture content is kept less than 12%. Whereas if the moisture content is more, so shelf life will be obviously less. And wet DDGS, wet distillers grain, which is available at distillers level, is having shelf life not more than 48 hours. So that we need to educate the farmers. Sometimes with enthusiasm, nutritionists say that DDGS is excellent and all those things, but we fail to pay attention to the minor details, what farmer is thinking. Farmer may be getting only wet DDGS. So wet distillers grain, obviously we have to say that it has to be consumed as early as possible. So that is the one thing.
Yes, sir. Another
So,
question we have, that any other DDGs other than maize and rice?
yes, in Europe, if you see the literature, it is conventionally they have wheat DDGs. Whereas as per the policy of Government of India, wheat is primarily a human food in not supposed to use wheat for animal feed production except broken wheat or etc., which is available through auction in FCI godowns etc. So that is why we even there are some report of sorghum DDGs available, but practically we have not seen because mostly today rice DDGs and maize DDGs are increasingly used, and government policy is also to push more and more maize towards ethanol manufacturing, resulting in increased DDGS availability for livestock feed usage.
Yes, sir. Another question is that, is it feasible to reduce the use of grains for ethanol production by incorporating alternative feedstocks like bamboo or sugarcane and ensuring minimal impact on the livestock feed industry?
Yes, so traditionally people were using sugarcane for ethanol manufacturing, but most of, as we, if you see the recent 5 years trend, most of the sugar mills are in the their growth is, it is rather we can say that they are degrowing in India. That is one thing. And so policy-wise, people say that even Niti Aayog report, if you read, the primary rationale for promoting these grains for ethanol production is overall reduced water footprint as well as reduced carbon footprint. So that is the one reason why they are pushing more and more grains compared to, and probably grain may be yielding more ethanol per unit of substrate compared to other feedstocks. So that could also be one of the possible reasons why Government of India is promoting more and more grains for biofuel production.
Thank you, sir. Thank you so much. And we are done with the question. Thank you for answering all the queries of the participants and satisfying them with your answers. Thank you so much. Your presentation was indeed very wonderful, and thank you for being the amazing speaker of today's session. Thank you, sir.
Thank you, Dr. Mahesh. Fantastic presentation. Very informative.
Thank you
I
very
also
much.
thanks Dr. Mahesh for accepting my invitation to express their view and expertise in this particular group of the audience. So definitely, I think all field veterinarians and all faculties related to the veterinary science and feed industry are benefited with this presentation, because it is not newer feed ingredient, but traditionally we are using this one to reduce the cost of feed nowadays whatsoever is increasing day by day. So definitely all stakeholders are understanding the use of the DDS today. Thank you, Mahesh, once again on the behalf of
Thank
our
you.
organizing
Thank
committee
you.
and college.
Thank you, Dr. Dinesh.
So, dear participants, as we today we have reached to the end of the session. So, on the behalf of Biotech Agriculture Forum for Technical Education of Farming Society Kota, Faculty of Veterinary Sciences and Animal Husbandry Ranchi, Birsa Agriculture University Ranchi, and the entire organizing team, I deliver my heartfelt gratitude to our amazing speakers, Dr. Pankaj and Dr. Mahesh, for their insightful talks. I'm very sure that their intellect and their vast experience must have motivated and boosted the knowledge of our participants. And from the response of the participants, which we got in the chat box daily, I think there is a need to organize many more trainings like this. And this is my humble request to the organizers to organize more and more such programs for veterinary professional. I think they are much interested in such training programs for increasing their knowledge and for getting the networking opportunities with the experts of their field. So I feel them, and I request the organizer on the behalf of the participants to get more and more experts on board, and to give them the chance for networking with the experts. So last but not the least, I would like to thank the participants for joining this 7 days training program and being so patient and attentive throughout the today's session, and asking their queries, and maintaining the decorum of the training sessions. So thank you, dear participants. Thank you, the organizer. Thank you, the speakers. So thank you all, and have a great evening.
Thank you all.